Welcome to this lecture on reproductive biology. Today, we are going to explore the complex and deeply impactful topic of male and female infertility. We will uncover the specific biological roadblocks that prevent natural conception, understand the fascinating mathematics behind pregnancy chances, and learn exactly how medical professionals diagnose these issues. Let us dive in. To begin our journey into reproductive biology, imagine you are trying to grow a beautiful garden from scratch. To get a healthy plant, you need several things to work perfectly together. You need a high-quality seed, nutrient-rich soil, a clear path for water, and the perfect weather conditions. Human reproduction is remarkably similar to this delicate gardening process. The seed represents the male sperm. The soil is the female uterus. The clear path refers to the open fallopian tubes. And the weather is the precise hormonal balance required for ovulation. If even one of these elements is missing or damaged, the garden will simply not bloom. Infertility is the medical term for when this natural process faces a roadblock. While the garden analogy gives us a conceptual understanding, in the medical world, we need strict guidelines. Infertility is formally defined as the inability of a sexually active, non-contracepting couple to achieve a clinical pregnancy after 12 months of regular intercourse. For women over the age of 35, this diagnostic window is shortened to 6 months because female reproductive potential naturally declines with age. It is crucial to understand that infertility is not a single disease. It is a complex clinical presentation that can stem from the male, the female, or a combination of both. Understanding this is profoundly important because it affects approximately 1 in 6 couples worldwide, causing immense psychological and financial strain. Now that we know the clinical timeline, we must first look at how likely a healthy couple is to get pregnant in a given time frame. Biologists use a concept called fecundability to measure this biological efficiency. Fecundability is defined as the mathematical probability that a single menstrual cycle will result in a clinical pregnancy. Because human reproduction is naturally quite inefficient, even perfectly healthy couples do not have a 100% chance of conceiving in 1 month. By understanding this baseline probability, doctors can predict how long it should normally take a couple to conceive, which mathematically justifies why we wait 12 full months before diagnosing infertility. Now that we understand the concept of fecundability, let us look at the mathematical formula used to predict conception. The formula is P sub n equals 1 minus the quantity 1 minus f raised to the power of n. Conceptually, this formula calculates the probability of failing to get pregnant every single month and subtracts that total compounding failure rate from 100%. Let us break down the symbols. P sub n is the cumulative probability of achieving a pregnancy after a certain number of months. The variable f represents fecundability, which is the chance of success in just 1 cycle. Finally, n is the number of months the couple has been trying. Physically, if the single cycle probability f increases, the cumulative success rate shoots up quickly. If n increases, meaning they try for more months, their overall chances also improve. Before we move on, let us see this formula in action with a concrete example. Suppose a healthy young couple has a normal fecundability rate of 25% per cycle, and we want to find their chances of conceiving after 6 months. We are given f equals 0.25 and n equals 6. We chose this formula because it perfectly models repeated, independent biological events. First, we substitute our values into the equation. Inside the parentheses, 1 minus 0.25 gives us 0.75. This is their 75% chance of failing in any single month. Next, we raise this failure rate to the power of 6, which gives us approximately 0.178. Finally, subtracting this from 1 leaves us with 0.822. Physically, this means out of 100 healthy couples, about 82 will successfully conceive within 6 months. This formula we just used leads us to an important clinical question. What happens when biological capacity is reduced? Let us look at a couple with subfertility, where a mild male factor issue drops their fecundability to just 5% per cycle. We want to calculate their odds over a full year, so F is 0.05 and N is 12. Plugging these into our equation, we find a massive 95% failure rate per month. Raising 0.95 to the 12th power gives us 0.540. Subtracting this from 1 yields 0.460. Physically, this means they only have a 46% chance of pregnancy after 12 months. Because they are more likely to fail than succeed after a full year, they officially meet the clinical definition of infertility and would benefit from medical intervention. Now that we understand the mathematics of why infertility is diagnosed after a year, we must explore the biological causes, starting with the male. Male infertility is primarily driven by issues with the production, quality, or delivery of sperm. The most common issue is oligozoospermia, which is a clinically low sperm count. If there are not enough sperm in the ejaculate, the statistical chances of even one surviving the long, harsh journey to the egg drop drastically. This condition can be caused by genetic defects, hormonal imbalances, or lifestyle factors like smoking. Without a sufficient starting population of sperm, the biological math simply works against the couple.
Beyond just the sheer number of sperm, we must also consider their physical capabilities. Another major issue is asthenozoospermia, which refers to poor sperm motility. Sperm must be excellent swimmers to navigate through the cervix, the uterus, and into the fallopian tubes. If they swim in circles or do not move at all, fertilization cannot occur. Finally, we have teratozoospermia, which refers to abnormal sperm morphology. This means the sperm have misshapen heads or tails, preventing them from properly penetrating the egg's tough outer shell. Even with a high sperm count, poor motility or abnormal shape will act as a complete roadblock to natural conception.
Having covered the male factors, we now turn to female infertility, which is often more complex because the female reproductive system has to manage ovulation, fertilization, and gestation. The leading cause of female infertility is ovulatory dysfunction, such as polycystic ovary syndrome. In these conditions, the hormonal weather is disrupted, meaning the ovaries fail to release a mature egg regularly. Without an egg, conception is biologically impossible. This disruption in the delicate hormonal sequence is one of the most common reasons couples struggle to conceive, but fortunately, it is often highly responsive to medical treatments that help regulate the cycle. In addition to ovulation issues, we must also look at the physical pathways and the environment of the uterus. A major cause is tubal factor infertility, where the fallopian tubes are blocked or damaged, often due to past infections like pelvic inflammatory disease. If the tubes are blocked, the sperm and egg are physically separated and can never meet. Additionally, conditions like endometriosis, where tissue similar to the uterine lining grows outside the uterus, can cause severe inflammation and scarring. Finally, age is a critical factor. As a woman ages, both the quantity and the genetic quality of her remaining eggs decline significantly, which naturally lowers her monthly fecundability. Now that we have explored the specific causes, let us visualize these biological roadblocks. On the screen, you are looking at a side-by-side schematic of the male and female reproductive systems. On the left, notice the red markers in the male system. One highlights the testes, representing low sperm production, while the other shows a physical blockage in the vas deferens, preventing sperm delivery. On the right, the female system shows three critical failure points. The marker on the ovary represents anovulation, where no egg is released. The marker in the fallopian tube shows a tubal blockage, physically separating the sperm and egg. Finally, the marker in the uterus indicates implantation failure. This diagram teaches us that reproduction is a sequential chain, and a break at any single point results in infertility. As we bring this lecture to a close, let us review the most critical points we have covered. First, infertility is a highly logical medical condition, defined by the inability to conceive after 12 months of regular intercourse. Second, by using the mathematics of fecundability, we clearly saw how small biological deficits compound over time, drastically reducing a couple's chances of pregnancy. Finally, whether the root cause lies in male factors like poor sperm motility or female factors like blocked fallopian tubes, the end result is a disruption of the delicate reproductive sequence. You now have a solid understanding of these biological roadblocks, which is the crucial first step before exploring modern assisted reproductive technologies.